I mean, it's probably the oldest story we have.
It's woven into everything.
For centuries, it was just that, a myth. You had explorers chasing these immortality elixirs, alchemists trying to, you know, create endless life.
Right, looking for that one magical spring.
It was pure fantasy. But now, it's different.
It's very different.
We're not chasing magic anymore. We're using molecular biology, and frankly, the results sound even more impossible than the old myths.
Yeah, you have real scientists in real labs doing these incredibly sophisticated experiments.
With stem cells, gene therapy, even blood manipulation, but especially with something called cellular reprogramming.
And the goal has shifted. It's not just about slowing aging down.
No, it's about actively reversing it, turning the clock back on old cells.
Hi, and welcome to Brain Binge, the show where we dive way too deep into the weird side of reality. Whether you're trying to fall asleep, stuck on a late-night drive, cleaning your room, or just need something interesting in the background, thanks for hanging out with us. Tonight we're asking, can we actually turn back the biological clock, not just slow aging, but reverse it?
And that really is the mission of this deep dive. Because for decades, medicine was all about treating age-related diseases one by one.
Right, you get heart disease, we treat the heart. You get Alzheimer's, we treat the brain.
Exactly. It's a very reactive approach. But the thinking has profoundly shifted.
How so?
The goal now is to go after the underlying damage itself, the fundamental processes that drive all of those diseases at the same time.
So instead of patching the leaks, you're trying to fix the pipes.
That's a perfect analogy. And the biggest tool in this new toolkit, the one we're really focusing on today, is cellular reprogramming. We're trying to fix the cell's actual operating system. Okay, so let's start there. To even talk about reversing aging, we have to agree on what aging actually is at a molecular level.
Yeah, it's not one single thing.
It's more like a cascade of failures, right? A whole system of things going wrong. Modern biology calls them the hallmarks of aging.
Precisely. And while there are a few, we can really focus on 5 key ones that are the main targets for reversal.
Okay, what's the first one?
The first one is where it all starts,
Oh.
the instruction manual. We call it genomic instability.
So, damage to our DNA?
Constant damage. Our DNA is getting hit by environmental factors, by errors when it copies itself, you name it.
And our cells have repair systems for this, right?
Incredible ones. I mean, a healthy cell might get, what, tens of thousands of DNA lesions every single day, and it fixes most of them.
But not all of them.
Not all of them. And over time, the repair system just gets fatigued, the errors start to pile up, and the cell starts to misbehave.
Okay, so that error buildup connects to the next hallmark, which is sort of like a built-in timer, right? Telomere shortening.
Exactly. Yeah. Telomeres are those little protective caps on the ends of our chromosomes.
Like the plastic tips on a shoelace.
Perfect. Every time a normal cell divides, those caps get a little bit shorter. They're sacrificial DNA.
And when they get too short?
The cell sees it as a crisis. It stops dividing permanently. It's like a cellular odometer hitting its limit.
So we have errors in the code and a limit on how many times we can copy the code. What's next?
Next is how the cell reads the code. This is a big one. Epigenetic alterations.
Epigenome.
Right. These are chemical tags, like methylation marks that sit on top of your DNA. They don't change the sequence, but they tell your genes when to be active and when to be silent.
So it's the software that runs the DNA hardware.
Yes. And as we age, that software gets messy. The carefully managed patterns get disorganized. Genes that should be off might turn on, and vice versa. The cell just loses its ability to read its own instructions properly. This is what we call epigenetic drift.
And that drift is what brings us to the zombie cells.
Ah, yes. Cellular senescence.
These are cells that are so damaged they've stopped dividing, but they also refuse to die.
They refuse to die gracefully, which is the key. Instead of just clearing out, they stick around and start screaming.
Screaming how?
Chemically. They secrete this nasty inflammatory cocktail of proteins called the SASP.
The senescence-associated secretory phenotype.
That's the one. And this toxic mix spreads inflammation to all the healthy cells around them. It's how localized damage becomes a systemic problem, driving things like arthritis and heart disease.
They're basically bad influences on a cellular level.
The worst. And that brings us to the 5th key hallmark, which is that the body's repair crews start to get worn out. Stem cell exhaustion.
So our ability to regenerate just fades.
It does. Our tissues rely on these pools of specialized stem cells to replace worn-out skin, blood, muscle. But over time, those pools shrink, or the cells in them just don't work as well. Recovery gets slower.
Okay, so if we put it all together, DNA errors, fraying chromosome caps, messy software, zombie cells poisoning everything, and tired repair crews. That's aging.
That is the modern, actionable definition of biological aging.
Which brings up a crucial point. If we're going to reverse this, we need a way to measure it. We need a ruler.
We do. And that's where the difference between chronological age and biological age becomes so important.
Chronological is just years on a calendar.
Right. Biological age is the true functional age of your tissues. And the tool that lets us measure that and prove age reversal is the epigenetic clock.
The clock developed by Steve Horvath.
Yes, at UCLA back in 2013, he discovered that those methylation patterns we talked about, the chemical tags, change in an incredibly predictable way as we age. It's consistent across almost all our tissues.
So he found a set of bookmarks on the DNA that act like a universal timekeeper for the body.
Precisely. You can take a skin cell from a 60-year-old, run it through the clock, and it will read about 60. But the exciting part is if you do something to that cell, an intervention, and the clock then reads 40.
Then you have objective proof that you've rolled back its biological age.
You do. You have a quantitative measure of rejuvenation.
But I have to ask the obvious question here. Is the clock measuring something that causes aging, or is it just correlated with it? Is it the driver, or just a passenger?
That is the critical distinction. The very first clocks were mainly correlational. But the newer clocks, like the Grimmage clock, are different.
How so?
They incorporate methylation sites that are known to be linked to things that drive poor health outcomes, like smoking or chronic inflammation. When an intervention moves that clock backwards, and you also see an improvement in actual physical function.
Then the case gets a lot stronger that the epigenome is a key driver of the whole process.
It does. The clock is the ruler that proves the other tools are working.
Okay, so with our ruler in hand, let's talk about the most radical tool of all, cellular reprogramming.
Right. To really get this, we just need a quick refresher on how flexible a cell can be. Its potency.
This is the hierarchy, from most powerful to least.
Exactly. At the top, you have two tipitin cells, like a fertilized egg. It can become the entire organism, placenta and all.
Then pluripotent.
Pluripotent cells can become any cell in the body itself. Think embryonic stem cells. And then you have multipotent cells, like a blood stem cell, which can form several related types of cells, but not everything.
And for a long time, the thinking was that once a cell specialized, once a skin cell became a skin cell, that was it. The decision was final.
It was considered a one-way street. That dogma was completely shattered in 2006.
By Shinya Yamanaka in Japan.
Yes, at Kyoto University.
Yeah.
This discovery is really the ground zero for modern age reversal science. It's a Nobel Prize-winning breakthrough.
His experiment was just so elegant. He took adult skin cells from mice.
Fully specialized cells.
And he inserted just 4 specific genes into them using a virus.
The famous 4, OCTO, SOX2, KLF4, and C-Mike, now known as the Yamanaka factors, or OSKM.
And the M in that C-Mike is the accelerator, right? It's a known cancer gene.
A powerful one. It forces the cells to grow and divide aggressively. And when you turn on all 4 of these switches at once,
the adult skin cell just reverted. It erased its identity.
Completely. It jumped all the way back up the ladder to become an induced pluripotent stem cell, or iPSC. It was pluripotent again, like an embryonic stem cell.
Which means the epigenetic clock on that cell was reset all the way back to 0.
Wiped clean. It proved that the marks of aging aren't permanent. The youthful programming isn't lost, it's just hidden under layers of epigenetic noise.
So this is incredible for making new tissues in a lab, but there's a huge problem when you think about using this for whole body rejuvenation.
A massive problem.
Yeah.
You cannot safely use full, complete reprogramming in a living person.
For 2 big reasons, right? First is the loss of identity.
Yes. If you reset a liver cell all the way back, it forgets it's a liver cell. It becomes a primitive, useless cell in the middle of a complex organ.
It's biological chaos.
And second, and much worse, is the lethality.
The tumors.
The tumors. If you express OSKM continuously in a living animal, that C-Mike factor goes wild. You get uncontrolled growth. You get these bizarre tumors called teratomas.
Which are tumors made of a mix of different tissues like hair and teeth.
It's chaos. Or you just get aggressive cancer. The animal dies.
So you have a button that rewinds time, but if you hold it down too long, it kills the organism.
Exactly. Which means the scientific challenge completely changed. It went from can we reset age to can we control the reset button safely?
And that leads us to the next big idea, partial reprogramming.
The middle path.
The idea of just tapping the reset button, not holding it down. turning the OSKM switch on just long enough to get the benefits, but not long enough to erase the cell's identity.
To find that perfect dial setting. And the first really big demonstration of this in a living animal came out of the Salk Institute in 2016.
This was the work with the progeria mice, the ones that age incredibly fast.
Yes, which gives you a very quick and clear result. The key to their experiment was the control system they used.
They didn't just inject the factors.
No, they engineered the mice, so the OSKM genes would only turn on when the mouse was given a specific drug, an antibiotic called doxycycline.
So they had an on-off switch?
A precise one. And instead of leaving it on, they pulsed it. 2 days on, 5 days off, then repeat.
That rhythm is the key.
It's everything. It gave the cells just enough of a taste of rejuvenation to start cleaning up their epigenome, but not enough time to forget who they were or start growing uncontrollably.
And the result in these fast-aging mice?
They were stunning. They saw systemic rejuvenation. Tissues looked younger. They functioned better. Pancreas, spleen, muscle. Regeneration improved. And most importantly, the mice lived about 30% longer than the untreated ones.
And the mice that got the continuous full-blast OSKM?
They died quickly, as expected. It proved that the window for safe rejuvenation is incredibly narrow, and that pulsing rhythm seems to be the secret.
Okay, so that's a systemic effect. But then David Sinclair's lab at Harvard took this a step further, targeting a very specific tissue.
A really challenging one. The eye. The central nervous system.
And their approach was even more focused on safety, wasn't it?
It was. They used a modified recipe. Instead of OSKM, they used just OSK.
They dropped the M.
They dropped c-MYC, the most dangerous cancer-causing factor, from the mix. And they delivered these OSK genes with a viral vector directly to the retinal ganglion cells in mice.
These are neurons in the eye, and neurons don't regenerate?
Notoriously so. Once they're gone, they're gone.
So what happened?
The results were, again, astonishing. In mice with age-related vision loss, or even with a crushed optic nerve, they saw a partial restoration of vision. They saw actual regrowth of nerve fibers.
That's not just slowing aging, that's reversing damage that was thought to be permanent.
And critically, when they checked the epigenetic clocks in those neurons, they had rolled back. The cells were biologically younger, but they were still neurons. They hadn't lost their identity.
Which gives a lot of weight to this idea of a youthful backup being stored in the cell.
Exactly. The theory is that the perfect youthful instructions are never actually lost. They're just archived.
And aging is just the noise, the epigenetic drift that keeps the cell from being able to read that backup file.
And partial reprogramming is like running a defrag program. It cleans up the noise and restores access to the original clean operating system, all without wiping the hard drive.
The challenge now is figuring out how to run that program safely across an entire human being without causing a catastrophic system crash.
That is the multi-billion-dollar challenge.
So reprogramming is the high-risk, high-reward strategy, but there are other approaches attacking the aging hallmarks that are, well, maybe a bit closer to reality.
Yes, for sure. Instead of trying to rewind the clock inside a cell, some strategies just focus on taking out the trash.
Getting rid of the zombie cells.
Exactly. Targeting cellular senescence. The goal is to find drugs called senolytics that can selectively kill these senescent cells without harming healthy ones.
And this works really well in mice.
Beautifully.
Yeah.
Their early studies from places like the Mayo Clinic show that if you clear out these cells from old mice, their health span improves dramatically. Better heart function, less frailty, they just age better.
And how do they work? How do you kill one specific type of cell?
You exploit your weakness. To avoid dying, senescent cells have to activate these powerful pro-survival pathways. Senolytics are drugs that block those specific pathways.
So you're pulling the plug on their life support.
That's it. You're basically convincing the zombie cell to finally undergo apoptosis, or programmed cell death.
The classic combo was dasatinib and quercetin. But how is this playing out in humans? That's what really matters.
And the human results have been mixed. It's much more complex in people.
There's not a magic bullet.
Not yet. There have been some early trials for specific diseases, like lung fibrosis or diabetic kidney disease, where senescence is a big driver.
Yeah.
And while some trials show the drugs are clearing out senescent cells, the big clinical improvements haven't always followed. It's a promising field, but long-term safety and efficacy in humans is still a big question mark.
Okay, let's switch to another hallmark, telomeres. If the problem is the caps on our chromosomes are getting shorter, why not just lengthen them?
But the enzyme telomerase, it's the most direct solution, in theory.
But also the most dangerous, right? Because cancer.
Because cancer. Over 90% of human cancers have reactivated telomerase to achieve their own kind of immortality. So by turning it on everywhere, you're potentially disabling a key anti-cancer mechanism that evolution gave us.
So we're back on that safety tightrope.
Always. But there was a fascinating study in 2012 from Maria Blasco's group in Spain. They tried to walk that tightrope.
How?
They used a controlled gene therapy to activate telomerase in adult mice, but not to an insane degree. And they found it did improve health and extend lifespan without a catastrophic increase in cancer.
So it's a powerful lever, but you need incredibly fine control over it.
Extremely fine control. It's not a switch you just want to flip on and leave on.
The last tool is a bit weirder. It's just from the cell to the whole system, the idea of parabiosis.
Ah, yes. The young blood experiments.
This is where they surgically join the circulatory systems of a young mouse and an old mouse.
Right. And the initial findings were pretty stunning. The old mouse started showing signs of rejuvenation, better muscle repair, better liver function.
Which led to this whole vampire therapy narrative.
It did. But the science gave more nuanced because the young mouse that was hooked up to the old mouse actually started aging faster.
So it wasn't just magic young factors flowing into the old mouse.
It seems it's more about the bad stuff in the old blood getting diluted. The old plasma is full of pro-aging, pro-inflammatory signals. By joining with the young mouse, you're essentially giving the old mouse a plasma exchange. You're diluting the old signal.
You're changing the systemic environment.
And that shows that aging isn't just happening inside the cell. The environment the cell lives in, the signals it gets from the blood, is profoundly important.
Okay, we've gone from reprogramming epigenetic clocks to clearing zombie cells to diluting old blood. It's an amazing picture of what might be possible.
It is.
But now we have to connect this back to reality, to the state of human medicine right now.
This is the most important reality check. We have to be crystal clear. True age reversal reprogramming is still preclinical. It's experimental. It's in mice.
There are no approved whole-body human rejuvenation treatments using this technology.
None. Full reprogramming is still incredibly dangerous. The gap between reversing blindness in a mouse eye and safely treating a whole 75-year-old person without causing cancer it's monumental.
And yet there's so much money pouring into this. Companies like Altos Labs, Calico, they're staffed with Nobel laureates.
And that's because they are tackling that monumental engineering problem. They're doing the foundational science to figure out how to make this safe. They're not making consumer products.
We have seen a few tiny human trials that gave some hints, though, right? The Triumph trials.
Yes, very small studies aimed at regenerating the thymus gland, which is key for our immune system.
And they used a cocktail of drugs, including growth hormone.
They did, and they saw some thymus regeneration, which was interesting. But the really fascinating part was they saw a small but measurable rollback on the participants' epigenetic clocks.
But these were, what, a dozen people?
Not even. And the use of growth hormone comes with its own list of risks. It's a proof of concept that the human epigenetic clock can be moved. It's not a longevity treatment you can go get.
So if reprogramming is in the future, what about the drugs people are already talking about? Metformin, rapamycin, NAD plus boosters?
They are definitely the most promising things we have in the near term, but we're still waiting for definitive proof of general human longevity.
Let's start with metformin. It's a cheap diabetes drug.
And some data suggest that diabetics taking it actually live longer than healthy people who aren't. It seems to work by activating a pathway called AMPK, which basically tells the cell it's low on energy and should switch into a repair mode.
And there's a big trial happening now to test it in non-diabetics.
The TAME trial, yes, but we're still waiting for those results.
And rapamycin.
Rapamycin is even more powerful in animals. It inhibits a central nutrient sensor called mTOR. It basically tricks the body into thinking it's fasting, which kicks off a massive cellular cleanup process called autophagy.
But it's also a powerful immune suppressant.
Which is a major side effect. So the risk-benefit for a healthy person is still very much an open question.
So we're left with the unsexy truth, aren't we?
We really are.
While we talk about all these high-tech interventions, the most powerful tools to move your epigenetic clock right now are the boring ones.
The boring but real levers. Consistent exercise improves mitochondrial health and reduces inflammation. A healthy diet does the same. Not smoking. Managing stress.
And maybe the biggest one of all, quality sleep.
Absolutely. During deep sleep, the brain runs its own waste-clearing cycle. It's flushing out metabolic junk. It is probably the single most powerful anti-aging intervention that currently exists, and it's free.
It's the ultimate irony. Scientists are racing to invent a molecule to reprogram your cells, and the best thing you can do is just go to bed on time.
For now, yes.
Okay, so let's imagine for a moment we solve the science. We find the safe way to do this. We still have to confront the biggest barrier of all.
The one that's always there. The link between reversing aging and causing cancer.
They seem to be two sides of the same coin.
They really are. The pathways you need to activate for rejuvenation, cell growth, cell division, are the exact same pathways that cancer hijacks.
Evolution designed those pathways to be shut down in older people for a reason.
As a safety mechanism. To prevent tumors. When we try to force them back on in a 70-year-old body that's full of accumulated DNA damage, we are playing with fire.
The more we learn to rewind biology, the more we realize how clumsy our hands are on the controls.
It's true. And it makes you wonder if this is really about extending health, or is it about being afraid for things to end?
And if we do solve it, if we get safe age reversal, the biological problem is immediately replaced by a much harder ethical one.
Immediately. The first question is access.
Who gets it? If this costs $1 million a treatment, what happens when only the rich can afford to stay biologically young forever?
You create a biological class system, something humanity has never seen before. A world of the biologically young elite and everyone else who ages and dies naturally.
It's one thing to live longer. It's another to decide who gets those extra years.
And what about society itself? Our entire social structure, retirement, careers, inheritance is built around a roughly 80-year lifespan. What happens if people are healthy and working until they're 120?
If you can update the age of your cells like an app, what does growing old even mean anymore?
It's a profound question.
Which brings up the most personal question of all. If you could safely reset your body to 25, knowing all this, would you actually do it?
That's the one everyone has to ask themselves. Because there's a psychological cost. What is it like to live in a 35-year-old body but have 150 years of memories?
Your mind would be ancient, but your body wouldn't.
Are you even the same person anymore? The burden of watching generations of loved ones pass by while you remain physically static. I'm not sure we're built for that.
So at the end of the day, the science is showing us that aging is malleable. It's a software problem that can, in principle, be fixed.
Demonstrated in cells, demonstrated in mice. But the path to making it a safe, equitable human reality is incredibly long. And the biggest questions aren't biological anymore. They're ethical.
The fountain of youth, it turns out, might just be a set of molecular switches in our own DNA. But having the power to flip them comes with a responsibility we are nowhere near ready for.
And until we are, the boring path of good sleep and healthy living is still the best medicine we have.
Thanks for listening to Brain Binge. Whether you're drifting off asleep, on a long drive, studying, or just getting things done in the background, we really appreciate you spending your time with us.
We do.
If you enjoyed this deep dive, please like, share, and subscribe, and tell us what you'd like us to explore next, or which part of the aging story messed with your head the most. Until next time, stay curious, stay safe, and have a blessed day.
That's it for this episode of Brain Binge. If you enjoyed getting your mind twisted tonight, do me a favor, comment, like, and subscribe so you don't miss the next deep dive. Got a weird fact, mystery, or place you want an episode about? Drop it in the comments. I read as many as I can, and I'm always hunting for new ideas. Thanks for listening, and thanks for letting your brain binge with me for a while. Stay well, stay curious, and I'll see you in the next one.